what did we do last time? Would you just remind me? Vagina, ovary, corpus luteum, we finished? Okay. So we continue with the female genital organs. This is the one. So today's slides are in the week 11A container. We start with the uterine tube, and we have two sections from this organ, which are different in the phase. So this 74 one is from the estrogen phase, and the other one, this 52-59, is from the progesterone phase. So let's start with the estrogen phase, the uterine tube, number 74. And this is going to be a cross-section with hematox and eosin staining. And the title says here uterine tube, but this organ has several other names. Could you name the other? Fallopian tube, oviduct, okay. In Latin, tuba uterina. That's something like uterine tube. Okay. So first of all, we can see two big lumens here. This is one of them, which is very labyrinthic. Actually, this is the cross-sectional profile of the uterine tube. This other similar size but much more regular lumen has nothing to do with the uterine tube. This is an embryological remnant. Let's start with this uterine tube. And because it is highly labyrinthic, the mucosa is highly labyrinthic, that's why the lumen, the border of the lumen is irregular. We can conclude that this sample was probably taken from the ampulla. This is one region of the uterine tube, for which this very labyrinthic appearance is characteristic. So this drawing is showing one of the uterine tubes as connected to the uterus, and the other end is freely opening into the abdominal cavity, and this is the so-called fimbriated end. These kind of fringe-like protrusions are called fimbriae, which can move and help in sweeping the ovulated egg cell into the cavity of the uterine tube. And along the length of the uterine tube, we can distinguish several parts. If we start from the uterus, the first part is called uterine portion, which is really very much connected to the cavity of the uterus. Then there is a narrow isthmus as we are traveling distally. And this part is called ampulla. This is the widest part of the uterine tube, and this is the most labyrinthic in its cross-sectional profile. So this is where the sample was taken from. And if you to think about the role of this uterine tube, obviously the egg cell, if it is swept into the lumen, has to be transported towards the uterus. At the same time, if sperm is deposited into the vagina, these sperm cells are coming from the opposite direction, through the vagina, through the uterus. They enter the uterine tube. So the direction of movement of the two egg cells are just opposite. And in most cases, the fertilization occurs here in the ampullary region. And the transport mechanism is not fully understood here, because sperm cells can move on their own, that's fine, but the egg cell cannot, so it has to be transported somehow, and this transport mechanism is essentially rely on movement of kinociliae, which are on top of the lining epithelial cells within the uterine tube, and also rely on the muscular components of the wall of the uterine tube, which can cause contraction of the tube. So we must assume that movement of cilia and these muscle contractions are in favor of the egg cell to get transported towards the uterine cavity. But if it is so, then it is against the movement of the sperm cell. So that's a kind of difficulty for the sperm cell to reach the egg cell. Now, this yellowish something is the ovary, from where we can expect the ovulated egg. And as I said, normally it is swept into the uterine tube. Sometimes this mechanism fails and the egg cell remains in the abdominal cavity, and it may get fertilized within the abdominal cavity, because through the open end of the uterine tube, sperm cells can enter the abdominal cavity. And if the egg cell gets fertilized somewhere in the abdominal cavity, then it starts its cleavage, the mitotic divisions. Morula is formed, later blastocyst is formed, and the outer cell mass, the trophoblast, differentiated into cytotrophoblast and syncytiotrophoblasm, and the syncytiotrophoblast, which is multinucleated syncytium, will synthesize proteolytic enzymes, whereby it can disrupt the nearby tissues to facilitate embedding. Now, the uterus is prepared for this, but elsewhere the abdominal cavity is not. So if this implantation starts somewhere in the abdominal cavity, it creates a danger for the mother and also for the implant, because this procedure, due to the presence of proteolytic enzymes, will disrupt the surrounding tissue and may cause internal bleeding of the mother, which can be life-threatening. So such a pregnancy outside the normal site, so the normal site of pregnancy is the uterus. So if pregnancy occurs outside the uterus, we talk about ectopic pregnancy. And you know, the most frequent site of ectopic pregnancy, most typical site of ectopic implantation is where? In the Douglas cavity or Douglas pouch or rectouterine cavity. So between the uterus and rectum, this is the lowest part of the abdominal cavity in female. So obviously this fact is related to the frequency of ectopic pregnancy there. Okay, and the other thing what you can see in this drawing is something called mesosalpinx. Actually, salpinx is the Greek name of the uterine tube. So an even further name, we already collected maybe four for this organ. And the mesosalpinx as a term refers to the duplication of the peritoneum, which reaches the organ. And this is, in terms of if you take the other similar term, mesovary, what we learned last occasion, this is quite similar, okay? And this is also visible in the section, the mesosalpinx. Well, the other one is better visible, but it is somewhere here. But if I open the other one, I can show you better. So the other uterine tube section, which is from the progesterone phase, this mesosalpinx is much better visible. Okay, so this area is the mesosalpinx, and obviously peritoneal coverage is here and also here, so eventually it surrounds the organ. This is an intraperitoneal organ, and this duplication is somewhere here. Okay, but first we just go back to the estrogen phase, the specimen. So this is the uterine tube cross-section in the ovular region. And if you look at the lining epithelium, it is simple columnar. And there are two kinds of columnar cells in this epithelium. One kind is paler, like this, and ciliated. It has kinociliae on top, so these ones. But there are also darker cells which are non-ciliated, like this, or like that. These are secretory cells, produce mucoid substance. So these are the two kinds of cells. I call your attention to the fact that the height of these two cell types are the same now in the estrogen phase. And I emphasize this because this will change in the progesterone phase. So that's one important thing on the basis of which you can recognize which phase the uterine tube is. Yes. Those are non-secretory cells, they are ciliated. The darker ones are not ciliated, but they secrete a mucoid substance. Okay, so the thing that these mucoid mucus-producing cells look dark, essentially basophilic, is probably that here the mucin was preserved, and mucin is a basophilic substance if it is preserved. Okay. And obviously the movement of the ciliae are important in the transport of the oocyte. Below we have the lamina propria, connective tissue, and there is no muscularis mucosae. Generally in the urogenital tract we don't have muscularis mucosae. So after the lamina propria, the external muscle layer comes, entirely smooth muscle, which can be divided into two sublayers. An inner circular sublayer and more or less outer longitudinal sublayer, so it's not so characteristic as the inner circular one, so it's difficult to demonstrate actually, but maybe in this territory you can see really cross-sectional profiles of smooth muscle cells, but you can see cross-sectional profiles of smooth muscle cells here as well. So these two sublayers are not very distinct, but usually consider that there is an inner circular and an outer longitudinal sublayer here. And the outermost tissue layer is subserosa and serosa. You can see some mesothelial cells here at the periphery. So this simple squamous epithelium is the serosa layer, and next to it there is some loose connective tissue, which is the subserosa. These are the layers. Now, this other big lumen is the lumen of the remnant of the mesonephric duct, which, remember, it is the excretory duct of the mesonephric system, which disappears or tend to disappear in females, but some remnants may persist even in females. In males, part of the mesonephric duct will differentiate into certain parts of the genital tract, but in females, most of it degenerates. But some remnants may be present, and one kind of remnant may be here, and this is called epophoron, okay, the 2O. So the first O is single, the second is double. Epophoron or parophoron. We cannot tell whether epophoron or parophoron. These are the cranial and caudal parts of those branches of the mesonephric duct, which in males differentiate into efferent ductals in the testis. But these degenerate mostly in females, but some remnants may be here. It is epophoron, if it is from the rather cranial part of this duct system, parophoron if it is more caudally. Epophoron or parophoron. There is another remnant which may persist in females next to the vagina. It is called Gardner's cyst. Gardner's cyst. In our vagina section we couldn't see it, but sometimes it is present next to the wall. Gardner's cyst. So these things are in your embryology book. If you look at the lining epithelium, it is something like simple columnar here. So it is much more regular let's compare to the progesterone phase. So let's go to this 52-59 section, which is the uterine tube again, but in the progesterone phase. Here you cannot see the embryological remnant in this specimen. This is also from the ampulla region, taking the fact that the mucosa is highly labyrinthine here. But if you enlarge, maybe somewhere here, yeah. If you look at the lining epithelium, it is still simple columnar, and we can see the two types of cells, the paler ciliated cells and the darker non-ciliated secretory cells. But the difference relative to the previously seen estrogen-phased uterine tube is that now these secretory cells are taller than the ciliated cells. And they are bulging, these secretory cells are bulging into the lumen. Okay, or here, or there. And because these secretory cells now have this characteristic shape which resembles the shape of a peg, something what you can use to hang a picture onto the wall, a peg, but you can hit it to the wall by a hammer, okay? So, because of the similar shape, these cells are called peg cells, But these cells are called peg cells only in the progesterone phase because they change their shape in the progesterone phase. In the estrogen phase, they don't have this kind of shape. So in that phase, we don't call them as peg cells, we call them just as secretory cells. These are still secretory cells, but in the progesterone phase, they are also called peg cells. And the reason for this relative change in height is that these cells are hormone sensitive to the estrogen and progesterone levels. And generally speaking, estrogen facilitates ciliogenesis, so formation of cilia, maturation of cilia. This happens during the first half of the cycle. And the most cilia and most mature cilia are present by the mid time of the cycle, so around the time of ovulation, when the oocyte can be expected and has to be transported within the uterine tube. As we discussed, cilia are very much needed for this, so this is biologically understandable. So this is what estrogen does. But progesterone, which dominates in the second half, will facilitate maturation of the secretory cells. So in this phase, somewhat more secretory product is expected to be deposited into the cavity of the uterine tube. So these cells are hormone sensitive. Yeah. Layers are the same, of course. So underneath we have lamina propria, then we have external muscle layer, and the outermost is subserosa and serosa. So this is subserosa. Well, serosa is not always visible because we tend to lose mesothelial cells, maybe one nucleus is here, but the other section we could see more actually. More than here. Here maybe one. Okay, so we tend to lose it. So that's it. Can we go on? So next we have two uterine sections. Again, one from the estrogen phase, the other one from the progesterone phase. So I'm going to open both because we need to compare them and learn how to find out which phase they are in. There are several features on the base of which we can decide. So I concentrate on the differences, therefore. Maybe I should project this slide first. On top you can see how two pituitary hormones, LH and FSH, are changing as a function of time during the ovarian cycle. And you can see that this dashed line represents the time of ovulation. And just before the time of ovulation, both hormones having a peak in their concentration. This is related to the timing of the ovulation. So here you can see diagrammatically that the egg cell is ovulated from the ovary. Here you can see a tertiary follicle. Okay? So this represents formation of maturation of follicles here in the first half of the ovarian cycle, and in the second half, this is represented here that corpus luteum is formed. Now, in the meantime, ovarian follicles and then later the corpus luteum will produce estrogen and progesterone. And you can see that in the second half, progesterone is dominating over estrogen, but this is just the opposite in the first half. And right below, you can see a schematic drawing of the mucosa layer of the uterus. Actually, we don't use this term mucosa in relation to the uterus. Instead, we use the term endometrium. So endometrium is nothing else than the mucosa of the uterus. And this mucosa is incomplete because there is no muscularis mucosae, okay? So endometrium consists of an epithelial layer and the lamina propria, but we don't use this term lamina propria in relation to the uterus. We will use other terms. But what you can see on the drawing is that there are changes in the thickness of this endometrium during the cycle. So here, 0 is when the menstruation starts, when part of the endometrial layer is lost. So the thickness is reaching a minimum by the end of the menstrual phase. And then the proliferative phase starts. It is called this way because cells will divide here in the endometrium and the matrix is renewed. So essentially the thickness of the whole endometrium will grow. And then it will decline again in sickness when the time of the next menstrual phase is coming. In the second half, we call it secretory phase in relation to the uterus, and this terminology is coming from the fact that there are uterine glands embedded into the endometrium. These are represented by these irregular lumens, uterine glands, exocrine glands, these glands start to produce lots of secretory product, mostly in the secretory phase, mostly in the second half of the cycle. So that's why the second half is called secretory phase, referring to the high amount of secretory product of these glands at this time. And all these things are of course interrelated, because estrogen and progesterone affects the cyclic changes in the endometrium. But this endometrium is not everywhere hormone sensitive in the uterus, because the uterus has several major gross anatomical parts. The major part is the body part. This upper convex part is what we call fundus. And this narrow barrel-shaped territory is called cervix. Now, the cervical part is not so hormone sensitive relative to the other parts. All sections are from the body part, probably, because it shows real hormone sensitivity. We see the differences between the two phases. Another important clinical kind of note here is that here the lumen at the territory of the cervix is called cervical canal, and it is connected to the lumen of the vagina. And we have seen that the lining epithelium of the vagina is 35 squamous non-keratinizing epithelium. But now we will see in a minute that the lining epithelium of the uterus is simple columnar epithelium. So there is a transition between the two types of epithelia, which is in the cervical canal. Also, in this territory, there is a change in the origin of the lining epithelium, because the lining epithelium of most of the vagina is from the cloaca, essentially, therefore endodermal. But the uterus is from the unification of the two mesonephric ducts, therefore mesodermal. So where the two kinds of derivatives meet, that's a danger zone, because these territories where derivatives of different germ layers meet are very prone to cancer. Okay? And sometimes there is a precancerous stage, which means that at the territory of the uterine, at the territory of the cervical canal, the epithelium start to change. Instead of having the normal simple columnar epithelium, which is characteristic for the uterus, it will become stratified squamous non-keratinizing. change in the typical lining epithelium when a new epithelium is being formed, a new kind of epithelium is being formed. This is called metaplasia. Metaplasia. Metaplastic changes may occur, and this is a precancerous stage. And this can be detected by routine checking of this territory by a trained medical doctor. Now, let's look at the section. So, if you compare both look like kind of male underwear. This is like a pant. This is also like a pant, but this is a big one and this is a small one. Okay, so these are the thighs here. Here also. Here is the testis. Now, it has a long history that medical students call this is a big pant, this is a small pant. And the big pant is the progesterone phase uterus, and the small one is the estrogen phase uterus. Well, if you are asked during the exam, why do you think that this or that is estrogen or progesterone phase, please don't say this. You may think about this, but you need to present some more biological reasoning, what I try to present next, okay? But for peace of mind, I just told you this story. So, so let's start with the estrogen phase or proliferative phase. No, this is not the one. The other one. So this more bluish territory here, this is the endometrium. Endometrium. This is the cavity of the uterus, of course. Endometrium. The mucosa of the uterus. Now, altogether, the rest is really something. This is the muscle layer of the wall. And in other organs we would call it as external muscle layer, but here we don't use this terminology. Instead we use myometrium as a name. So this muscular part of the wall is the myometrium. Entirely smooth muscle can be divided into 3 sublayers. But as you can see, there are artificially cut edges everywhere, because very regular. Unfortunately, the assistant who did this in the past, she believed that the rest is not important, so just dropped away. So we cannot see the entire myometrium. The outermost layer is missing, and also missing the so-called perimetrium. Perimetrium would be the subserosa and serosa in other organs, but here we don't use this term either. Instead we use perimetrium. Perimetrium is absolutely missing. And this is true for both uterus sections. So we can see part of the myometrium and the endometrium in both sections. The rest was lost. Now, if I enlarge the endometrium and look at the lining epithelium, well, it's not very, this is better here. So you can see simple columnar epithelium. And some cells are ciliated, not all of them, some are ciliated. Below, we have the rest of the endometrium. And these lumens are the lumens of the uterine glands. And the lumen is quite regular now, because almost no secretion is going on yet. So, very regular order. The endometrium itself is divided into two major sublayers. This more basal part, which is looking like dark hair, this is the basal layer, and this upper part, which looks paler and more loosely organized, this is the so-called functional layer. And it is called functional layer because during menstruation, this upper part of the endometrium was lost. But the basal part remains even during menstruation. Now, the fact that this functional layer sloughs off during menstruation, this is true for most of the wall of the uterus, but it is not characteristic for the cervix region because it is not hormone sensitive, and this degeneration and loss of the functional layer is related to the hormone sensitivity of certain cells here, but this is not the case in the cervical region. However, yes. Yes, but we can differentiate the functional layer into two further sublayers only in the progesterone phase, not now, because there is no way how we can differentiate here. Okay. The glands are exocrine glands, kind of tubular glands, which lead the secretum onto the surface. And these glands produce mostly glycogen and lipids. So why is it good biologically that the surface of the endometrium will be laid down by lipids and glycogen, the secretory product of these glands? Why is it important biologically in your opinion? No, not really. Protection against what? It could be, but well, this is the uterus, not the vagina. In the uterus, it is not so natural to have a microbiome. It is natural in the vagina. So, something else. It is related to the embedding process. When the egg cells arise, actually not the egg cell, because by that time it is blastocyst. So the blastocyst will start its embedding process. And there are different phases of nourishment during our life. And during this time when the blastocyst is being embedded, it has additional nourishing material because placenta is not functional yet. There is no placenta at this time yet. And the cytoplasm of the oocyte contains lots of nutrients, but by this time it is not sufficient because it has been used. So during this embedding process, the blastocyst takes up nutrients here from the tissue, which contains glycogen and lipid, which is created by the glands. Okay. The secretory mechanism of these glands is merocrine. And if you magnify one of these glands, maybe something here. So you can see that the nucleus of secretory cells is columnar, and the nucleus is more or less in the center of this columnar cell, right? This will change in the progesterone phase. Also the lumen, the border of the lumen is quite regular. This will also change in the progesterone phase. It will be much more irregular. Now the cells here in the endometrium are connective tissue cells because this is connective tissue. But in this organ, we call these cells as stroma cells. Stroma cells. Stroma cells collectively. Now, if you look at the myometrium, one feature here that there are so if you take a blood vessel here, because this myometrium is well vascularized. So let's take this blood vessel. In the wall there are muscle cells, smooth muscle cells of course, but what is quite unique that this tunica media of the vessel is very much indistinguishable from the rest of the myometrium. So the myometrium is generally full of smooth muscle cells, and these smooth muscle cells, this population of smooth muscle cells, continuously getting into tunica media of the blood vessel. That's a kind of characteristic feature here, that there is no sharp border between the tunica media of the vessel and the rest of the myometrium. Okay. Now, this black is something is a complete artifact, so don't ask what it is, I have no idea. It is just some dirt. Now, let's look at the other version. So during this time, what we have seen, the thickness of the endometrium is increasing. The minimum is about 2 millimeters, and it will reach something like 8 millimeters or even more when it is the thickest. And if you compare, you can really see that, well, I need similar magnification, okay, 5,000 micron, 5,000 micron. So it is more or less visible that here the endometrium is thicker than here. So let's magnify this progesterone phased, or also called secretory phased uterus. So first of all, we can distinguish the basal layer, or stratum basale, from the functional layer, or stratum functionale, just like before. But now you can further differentiate the functional layer into two sublayers, because here the more superficial part of this functional layer looks more compact and darker. Cells are closer to one another in this superficial part of the functional layer. layer than here in the deeper part. And the reason why this deeper part looks paler is mainly due to the fact that nuclei, cell nuclei, are farther apart. The nucleus is the darkest part of the tissue. If nuclei are farther apart, then the general impression will be that the tissue is paler. If the nuclei are farther apart, then the cells are farther apart. And the reason why they are farther apart is that something accumulates in the extracellular matrix which pushes cells away from each other. This something is a fluid which is derived from blood. And therefore this tissue is swelling, this part of the functional layer. It is full of fluid, just like a sponge can be full of fluid, okay? Because of this feature, this paler fluid-rich territory of the functional layer, which is the deeper part, this is called spongy layer. Spongy layer referring to the sponge, which can take up lots of fluid, just like this tissue. Spongy layer, and the upper darker territory is the compact layer, because cells are more compactly arranged here, therefore looks darker. Now, the process which is going on here in the spongy layer is edema. Edema when fluid accumulates in the extracellular matrix of a connective tissue and it is getting swollen. This is what we call edema. You have probably heard about it. You should have in first semester. In relation to which substance? We mentioned the cell which releases certain substance at this time. For example, during allergic reaction, which may lead to edema. Histamine, which is released by which cell? Mast cell. Okay. Now, this what we are recalling now, what happens or may happen during allergic reaction is not normal. Okay. But this edema here is normal. It is not an allergic reaction. So it happens in each cycle that this part of the functional layer, what we call spongy layer, gets edematic. That's normal here. Okay. So remember, in the first half, we couldn't differentiate the functional layer into compact and spongy layers. It is possible only in the secretory phase. Did you want to ask something? No, this is not really related to histamine release. Histamine is released during allergic reaction because during that time plasma cells, instead of producing IgG, they will produce IgE. And IgE will bind to IgE receptors on the surface of mast cell, and this will trigger the release of histamine. Now, this is not happening here.
Then what causes the edema in the progesterone phase?
Probably because blood vessels become leakier, but it is not due to the histamine, but probably due to hormonal changes. Now, I project the slide here, not this one, but this one, showing the endometrium and part of the myometrium here. And you can see a uterine gland here in yellow, and you can see some arteries here. And so glands have a portion in the basal layer, in the stratum basale, and the upper portion is located in the functional layer, okay? And although this drawing is not very good in this context, but the glands have a rather straight portion here in the basal layer, and a more spiraling part in the functional layer. And maybe return to this drawing later. So here in the basal layer, you can see that the glands have a really straight course, okay? They are rather straight. But here in the spongy layer, they are becoming curvy. Can you see? Curvy. Especially this one. So, like this, spiraling. And also, there are lots of secretory product in the lumen. This is not the case in the first half of the cycle, because at that time glands haven't really started producing the secretory product. And also, if you magnify, now you can see these secretory cells, which look a bit different now, because remember, in the first half of the cycle, the nucleus was centrally located inside the cell. But now you can see that the apical portion is bigger than this basal portion, because here in the apical part, secretory product is already accumulating, which pushes the nucleus down. Okay? So the location of the nucleus changes during the secretory phase. Okay? And they have a spiral course in the spongy layer. Okay, now I go back to the drawing. And here, from the side of the myometrium, artery is coming, which is actually branches of the uterine artery. And this branch has a rather straight course at the bottom part of the basal layer, like here. But in the upper part, mostly in the spongy layer, the artery becomes spiral shaped. Now, the spiral artery appears in the secretory phase, in the second half. During the first half of the cycle, in the estrogen phase or proliferative phase, still there is no spiral artery. So this is also a difference, that spiral arteries appear in the secretory phase. And I show you how you can recognize a spiral artery. Look at this. We are in the spongy layer, right? And this is a spiral artery here. What you need to look for is lots of profiles of arterioles next to one another. And this is the same spiral-shaped vessel which is crossed by the plane of sectioning, cut by the plane of sectioning many times. That's why we can see multiple sectional profiles, although we have just one spiral artery here. So this is the appearance of a spiral artery, okay? Which is actually an arteriole, but we call it spiral artery. And in the upper part of the spongy layer, or even in the compact layer, you can see these dilated vessels, blood vessels, which are called lacunae. spiral arteries appear in the second half of the cycle, lacunae also appear in the second half of the cycle. And something else is happening here in the compact layer. If we enlarge this territory, compact layer, in certain parts you can see very pale stained cells which have more or less basophilic cytoplasm and euchromatic nucleus, like this, or like that, or like that. Or maybe these ones here. Or even these ones even better. Where you see these very pale nuclei. These cells are a little bit bigger than other nervi cells. Now these paler, slightly basophilic and slightly bigger cells are derived from the stromal cells during the secretory phase. And this transformation is due to hormonal changes, due to the increasing progesterone as well. And these cells are undergoing a so-called epithelioid transformation. Epithelioid transformation, which means that these cells, remember they used to be stromal cells, which means connective tissue cells. And these connective tissue cells are becoming more and more similar to epithelial cells. This is what epithelioid transformation means. They are becoming epithelial-like cells, epithelioid cells. In that they can synthesize, accumulate and release certain substances in large amount. This is typical for glandular epithelium. Okay? Now these cells are called pseudodeciduous cells. Pseudodeciduous cells. Which means almost deciduous cells. They are not real deciduous cells as yet. They will become real deciduous cells only if implantation occurs. This is just in preparation for pregnancy. But real deciduous cells appear, so this epithelioid transformation is completed only if implantation occurs, so pregnancy occurs. We will see next time in the placenta section, in the pregnant uterus section, real deciduous cells, which are even bigger, even paler, even more basophilic than these pseudodeciduous cells. Now these cells accumulate glycogen and lipid, just like the glands of the uterus, glycogen and lipid. And the purpose is the same, to supply the implant with these nutrients during the embedding process. Okay? So this is something which is happening during the end of the secretory phase. It is definitely not happening in the proliferative phase. So if you can see pseudodeciduous cells, that's a sign that you are looking at the uterus in the secretory phase, right? Okay. So next, we need to talk about the menstruation. What kind of factors are involved in initiation of menstruation? So menstruation is nothing else than the loss of the functional layer where it is hormone sensitive, so not in the cervical region but elsewhere. But even during menstruation, the basal layer of the endometrium remains at place. So why is this difference? You would ask. Now the thing is that smooth muscle cells in the wall of spiral arteries are sensitive to progesterone level. But smooth muscle cells are sensitive to progesterone only in the wall of spiral arteries. Smooth muscle cells in the straight part of the artery, which is in the basal layer, it is insensitive to changes in progesterone level. Okay, so this is a fundamental difference which can explain why the functional layer is lost during menstruation, but the basal layer remains at place. And the explanation is coming now. So when progesterone level starts to drop towards the end of the cycle, because the corpus luteum starts to degenerate in the ovary, okay? So less and less progesterone will be produced by the degenerating corpus luteum. So progesterone level drops, and smooth muscle cells in the wall of spiral arteries will contract as a consequence due to the dropping progesterone level. So, but the straight part of the artery in the basal layer will not contract. So that's the fundamental difference. Now, I go back to the drawing. So imagine that here, in the spiral part, the wall is contracting. So blood flow will stop inside the spiral artery. It means that actually the functional layer will not get enough nutrients and oxygen, because from the spiral artery blood should flow on to reach the functional layer to supply those cells there with nutrients and oxygen. This is not happening because blood stopped flowing here. So cells in the functional layer will suffer from hypoxia, lack of oxygen, and they cannot get enough nutrients. Therefore they start dying, and eventually they die. So the tissue starts to degenerate, but only the functional layer starts to degenerate. The basal layer won't, because here in the basal layer we have the straight part of the artery which is not hormone sensitive, so it is not contracting. So blood can flow up to this point, can supply the basal layer with nutrients and oxygen. So those cells live further happily, unlike the cells of the functional layer. Now, the degenerating functional layer is not enough, because the wall of the spiral artery itself is degenerating, because in the wall of an artery, cells get nutrients and oxygen in the wall from the lumen. But no more fresh blood is arriving, so no more oxygen and nutrients is arriving. Therefore, the cells in the wall of the spiral artery will also degenerate. And eventually, the blood pressure will rupture the spiral arteries, because by that time the tissue of the wall has degenerated. So this is why and when bleeding starts, the menstrual bleeding, when the spiral arteries rupture. And this increased blood flow, as blood is escaping from the degenerated spiral arteries, will slough off the already degenerated functional layer. Okay? So it also means that within the menstrual flow, not only blood is found, but every other parts of the functional layer is present. So degenerated dead stroma cells, pseudodecidual cells, cells of the uterine glands. These are all present, the lining epithelium. These dead cells are also present in the menstrual flow. Okay, now as we stated, the basal layer stays, and therefore the endometrium can regenerate because the basal layer contains all the essential parts of the endometrium from which the whole endometrium with all its structures can regenerate. Because here we have the state artery which remains, it can further give rise to spiral arteries in the next cycle. The basal part of the gland also stays in the basal layer, the state part, and it will grow again in the second cycle. Okay? So gland can also regenerate. Of course, in the stratum basale we also have stromal cells which can proliferate and again give rise to the functional layer in the next cycle. So this is how regeneration can occur. So this was the story of menstruation. Unfortunately, many students cannot tell the story of menstruation, which is absolutely unacceptable, needless to say. Okay? Yes? Stromal cells proliferate to stromal cells, but later on they will differentiate into pseudodecidual cells.
Yeah, the pain and due to the endometrium being disturbed.
Well, you know, there are nerves here. I didn't mention, but you can imagine there are nerves, free nerve endings which are pain sensitive. So I can imagine it is painful. Ask the girls. We are the lucky ones, at least in this respect. Anything else? No. No. What you can see is the state part and the spiral. So this is the spiral part, and I think maybe here I could show you some state part. Okay. So, okay, we have still some minutes left. We essentially completed the material. As a repetition, I just would like you collectively to recollect the features on the basis of which we can differentiate between the two phases of the uterus. So, could you start? One feature on the basis of which you can differentiate between the two phases. No, uterus. Okay, presence of pseudodecidual cells in the compact layer. This is characteristic for which phase? For the progesterone phase or secretory phase, yes.
So being able to differentiate between the two layers of the functional layer, like the compact.
Yes, this is also characteristic for the secretory phase. Yeah, spiral arteries appear in the spongy layer in the secretory phase. Yes. Yes. Well, lacunae, yeah, I mentioned this. This is not so clear, but more or less, yes. Because if you enlarge here, we cannot really see these lacunae now in the proliferative phase. Yeah, okay. Anything else?
Thickness of endometrium.
Thickness of the endometrium, of course, is bigger in the secretory phase. Secretory cells of glands. What about them? The location is changing because in the secretory phase they are not centrally located but more basally shifted, because in the apical region the secretum is accumulating. In the proliferative phase the nucleus in central position.
Could we mention the staining of how in the secretory phase it's much darker and in the
No, I didn't say that. What I said that within the functional layer, the staining is different in the secretory phase, which is related to the edema in the spongy layer and the less edematic compact layer. So that's, yeah. But the shape of the gland, what you haven't mentioned. No, the gland, not the section. Yes, in the secretory phase, the lumen of glands gets more irregular, and also the secretum appears in large amount in the lumen in the secretory phase. No, the height is not really different, just the location of the nucleus. Maybe, but I wouldn't emphasize this. Okay, so is there anything left out, or we completed? What is it? Yes? Yes, you should know the names, but they are very difficult to differentiate the sublayers, but in theory you need to know. Yeah. Anything else?
Sir, you mentioned that in the uterine consists of progesterone and facilitates secretory cells to develop the complex. Does that also apply to secretory cells in the because like
Secretory cells of what?
in the uterus?
Well, obviously, they are hormone sensitive. That's why they start producing more secret. Yeah. Okay. Any more questions left?